Hi everyone, I am Dr. C. S. Mukrani, MD, and I am going to teach medical biochemistry. Before I start the topic, please like, share, and if you are new to the channel, please subscribe it so that the next video will be intimated to you. In the last session, we ended up with digestion of lipids. In today's session, we will continue with the same, in which bile and lipolytic enzymes are needed. Then we have digestion of triglycerides, in which there are short chain fatty acids, medium chain fatty acids, and long chain fatty acids. Good morning, everyone. Let us begin the topic. Digestion of lipids. Bile salts are important for digestion of lipids. The bile salts present in the bile, that is sodium glycocholate and sodium taurocholate, lower the surface tension. They emulsify the flat droplets. The emulsification increases the surface area of the particles for enhanced activity of the enzymes. In this diagram, we can see that bile salts emulsify the fat. Coming to lipolytic enzymes in intestine, they are pancreatic lipase with colipase, cholesterol esterase, phospholipase A2. The bile at pH 7.7 entering the duodenum serves to neutralize the acid chyme from the stomach and provides a pH favorable for the action of pancreatic enzymes. Here are certain physiologically important lipases. Lingual or acid stable lipase. Its site of action is mouth and stomach. Its preferred substrate is triacylglycerols with mono or short chain fatty acids. Its products are free fatty acids plus diacylglycerol. Pancreatic lipase plus colipase. Its site of action is small intestine. Its preferred substrate is triacylglycerols with long chain fatty acids. Its products are free fatty acids plus 2-monoacylglycerol. Intestinal lipase with bile acids. Its site of action is small intestine. Its preferred substrate is triacylglycerol with medium chain fatty acids. Its products are free fatty acids plus glycerol. Phospholipase A2 plus bile acids. Its site of action is small intestine. Phospholipid and preferred site substrate is phospholipid with unsaturated free fatty acids on position 2. Its products are unsaturated free fatty acid and isolecithin. Lipoprotein lipase plus insulin. Its site of action is capillary walls. Its preferred substrate is triacylglycerol in monochylomicron or VLDL, and its products are free fatty acids plus glycerol. Hormone sensitive lipase. Its site of action is adipocyte. Its preferred substrate is triacylglycerol in adipose tissue cells. Its products are free fatty acids with diacylglycerol or monoacylglycerol. Coming to digestion of triglycerides. Pancreatic lipase can easily hydrolyze the fatty acids esterified to the first and third carbon atoms of glycerol, forming 2-monoacylglycerol and 2 molecules of fatty acids. Then an isomerase shifts the ester bond from position 2 to 1. The bond in the first position is then hydrolyzed by the lipase to form free glycerol and fatty acids. Here we can see complete hydrolysis of triglyceride. Triacylglycerol is converted to 2,3-diacylglycerol plus R1COOH in the presence of enzyme lipase. Then this 2,3-diacylglycerol plus R1COOH undergoes 2-monoacylglycerol, which is done in the presence of lipase enzyme. Then this 2-monoacylglycerol is converted to 1-monoacylglycerol in the presence of lipase. Then this 1-monoacylglycerol is converted into glycerol plus R2COOH in the presence of enzyme lipase. The major end products of the digestion of triacylglycerol are 2-monoacylglycerol, that is 71%, 78%, 1-monoacylglycerol, that is 6%, and glycerol and free fatty acids, that is 14%. Thus, digestion of triglycerol is partial, that is incomplete. Cholesterol ester may be hydrolyzed to free cholesterol and fatty acids. The action of phospholipase A2 produces lysophospholipid and a fatty acid. In this diagram, we can see that triacylglycerol ethanolamine is broken down into small molecules by phospholipases. The binding of colipase to the triacylglycerol molecules at the oil-water interface is obligatory for the action of lipase. The colipase is secreted by the pancreas as an inactive zymogen, its molecular weight being 11,000. It is activated by trypsin. Coming to the absorption of long-chain fatty acids. Long-chain fatty acids, that is chain length more than 14 carbons, are absorbed to the lymph directly and not directly to the blood. Mixed micelle formation. The products of digestion, namely monoglycerides, long-chain fatty acids, cholesterol, phospholipids, and lysophospholipids, are incorporated into molecular aggregates to form mixed micelle. Here we can see the micellar formation. The micelle is formed by bile salts, which is red in color. Inside the bile salts, there are long-chain fatty acids and monoacylglycerol, and cholesterol is situated at the door of the circle. The micelles are spherical particles with a hydrophilic exterior and hydrophobic interior core. Due to their detergent action, the bile salts help to form micellar aggregates. Micellar formation is essential for the absorption of fat-soluble vitamins such as vitamin A, D, and K. The micelles are aligned at the microvillous surface of the jejunal mucosa. Fatty acids, 2-monoacylglycerol, and other digested products passively diffuse into mucosal cells. Here we can see that absorption of fat as chylomicrons. Fat brockets undergoing emulsification and micelle formation, together they come and form monoglycerides and fatty acids. Then these two diffuse into the mucosal cell. Then they leave the mucosal cell by triacylglycerol and lipoprotein form microns and finally enter the lacteals. Coming to enterohepatic circulation of bile salts. The bile salts are left behind, which are mostly reabsorbed from the ileum and returned to the liver to be re-excreted. This is called as enterohepatic circulation. About 98% of the dietary lipids are normally absorbed. Coming to re-esterification inside the mucosal cell. Once inside the mucosal cell, the long-chain fatty acids are re-esterified to form triglycerides. The fatty acids is converted into fatty acyl-CoA by the enzyme acyl-CoA synthase or thiokinase. This needs lysis of two high-energy bonds. Absorption of fatty acids. In this diagram, we can see that mixed micelles is broken into three parts. First part forms free fatty acids and 2-monoacylglycerol, which is converted into triacylglycerol, which is converted into chylomicron, which enters the lacteals. The second part is short or medium chain free fatty acids, which enters into the capillaries. The third part is released by bile acids, which is reabsorbed by ileum, which enters the portal vein and then into the liver. Two such activated fatty acids plus monoacylglycerol form triglyceride. Majority of molecules follow this MAG pathway. Free glycerol absorbed from the intestinal domain directly enter into the bloodstream. So the free glycerol is not available for reesterification, but the cells can convert glucose into glycerol phosphate and then add three molecules of acyl groups to form synthesized triacylglycerols. Coming to chylomicrons, the triacylglycerol, cholesterol ester, and phospholipase molecules, along with the apoproteins A and B48, are important incorporated into chylomicrons. The chyle, that is the milky fluid from the intestinal mucosal cells loaded with chylomicrons, are transported through the lacteals into the thoracic duct and then emptied into lymph circulation. In this diagram, we can see summary of utilization of fat. First, the fat from the liver is converted into VLDL with triacylglycerol in the blood, is sent into adipose tissue, mucosal and peripheral tissues, and finally into the lead to oxidation and generation of ATP. Second part includes from fat from the duodenum is hydrolyzed as triacylglycerol. It enters the adipose tissue, muscle and peripheral tissues and finally leads to generation of ATP. The serum may appear milky after high fat meal, that is postprandial lipemia, due to the presence of chylomicrons in circulation. Normally, the lymph clears within a few hours by the uptake of chylomicrons by tissues. Coming to short chain fatty acid absorption, which is different. Short chain fatty acids, that is SCFA, seen in milk, butter, and ghee, and medium chain fatty acids, that is MCFA, present in coconut oil and mother's milk, do not need reesterification. They can directly enter into blood cells, then into portal vein, and then finally into liver, where they are immediately utilized for energy. Their absorption is rapid. They are better absorbed than long chain fatty acids. Coming to abnormalities in absorption of lipids. Defective digestion in steatoia, daily excretion of fat is in feces is more than 6 grams per day. Greek word steer means fat. It is due to chronic diseases of pancreas. In such cases, unsplit fat is essential in feces. Defective absorption. On the other hand, if the absorption alone is defective, most of the fat in feces may be split fat, that is, fatty acids and monoacylglycerides. Defective absorption may be due to diseases like celiac disease, sprue, Crohn's disease, surgical removal of intestine, obstruction of bile acid, etc. This may be due to gallstones, tumors of heads of pancreas, and enlarged lymph nodes, lymph glands. The results of deficiency of bile acids. In such cases, triglycerides with short chain and medium chain fatty acids are digested and absorbed properly because they do not require micellization for absorption. Since milk fat and coconut oil are made up of MCT, that is medium chain fatty acids, they are therapeutically useful in malabsorption syndromes. Coming to chyluria, there is an abnormal connection between urinary tract and lymphatic drainage system of the intestine. Urine appears milky due to the lipid droplets. Coming to chylothorax, the Chirothorax can result from an abnormal connection between pleural cavity and thoracic duct. Here ends our session. Kindly like, share, and subscribe the channel. Thank you.